So I'm Johanna Swartz. I'm a postdoc at Duke University in the lab of Ahmad Hariri. And so I'm really excited to be speaking at Flux 2.0 and particularly excited that we're in LA because I'm from LA. So it was great to be able to get to return home for a conference. So I'll be talking today about results of a longitudinal fMRI study looking at changes in amygdala reactivity across adolescents as a function of risk for depression. Okay, so we're starting to form a relatively clear picture of what's happening in the brain of adult depressed patients. So this image is from a meta-analysis of fMRI studies that have looked at adult depressed patients compared to psychiatrically healthy controls. And the regions in red are the regions that are more activated in depressed patients compared to controls, specifically during negative emotional processing, so the processing of negative or threatening material. And what you can see in the middle there is that the limbic region and the amygdala as one of those structures is consistently hyperactivated in depressed patients compared to controls in adults during negative emotion processing. But one of the largest questions remaining unanswered is how does this pattern of neural activity develop? And so there are a couple of components of this question. So one is how does this emerge? So is this something that's present in some people from birth onwards, or does this emerge at some point later on in development? And if so, when does this pattern of functioning emerge and how does this emerge? And then also related to that is whether this pattern of functioning is something that we can observe prior to the onset of depression. So whether this is a premorbid vulnerability that might actually contribute to the onset of disorder, or maybe this heightened amygdala reactivity that we see in depressed patients actually emerged as a consequence of developing the disorder and all of the biased cognitive patterns that we know exist for depressed patients. So obviously we can't get at any of these questions by just looking at adults. And so this means we need to start looking at prospective longitudinal research. And a really interesting time to look at this is the period of adolescence, and Dr. Steinberg's talk was a great intro to this. So we know that adolescence is the peak period of onset for major depressive disorder. So this is a study following individuals who either had a family history for depression or no family history for depression. So the green line is showing individuals who had one or more parents with a history of major depressive disorder, and the purple line is showing individuals with no parents with major depressive disorder. And for both groups, the peak age at onset for incidence of depression was between 15 and 20 years of age. So this period towards the end of adolescence and the beginning of young adulthood looks like it's a period of heightened risk for the development of disorder. Adolescence is also interesting, given that it's this time of greatly heightened plasticity and change, and that lots of things are growing and developing during this period. But because it's a time of increased plasticity, this could also be a period when abnormalities in brain function emerge for the first time, potentially for some individuals. So we're starting to form a picture of what's going on in terms of typical adolescent development. So these are generally psychiatrically healthy youth that are functioning pretty well from cross-sectional research. So this is a figure from one of my papers, and this effect has been shown many times, that in general, amygdala reactivity decreases across the period of adolescence into young adulthood. So some studies have shown an inverted U-shaped effect where amygdala reactivity increases from childhood into adolescence and then decreases from adolescence into young adulthood. Others have shown a linear decline from childhood to adulthood. But what seems to be the consistent pattern across all these studies is this decrease in reactivity across adolescence into young adulthood. So we hypothesized that this is the typical developmental pattern that we should expect. People should be showing decreases in amygdala reactivity during adolescence. Then maybe individuals who go on to develop depression or who are at risk for depression fail to show these typical declines in reactivity with age. And as a result, they would have heightened amygdala reactivity at the end of this period. So we looked at this because we wanted to look at this prospectively before the onset of disorder. The way we got at this was looking at the effect of two major risk factors for depression on the development of amygdala reactivity during adolescence. And our risk factors were a family history for depression, which we defined as having both a first and a second-degree relative with a history of depression. So these individuals are at greatly increased risk of going on to develop the disorder themselves. So some studies suggest from their samples of adolescents with a family history that about 60% will go on to develop depression themselves. Other studies say even higher percentages. So it's probably safe to say that for the adolescents in our study who have a family history, the majority are going to go on to develop depression at some point. We also looked at the effects of stress, and we looked at childhood maltreatment as well as stressful life events occurring during adolescence. And we actually had objective ratings of life event severity. So the life event ratings took into account the severity of the life stress. So we know that these are also risk factors for the development of depression. And in addition to looking at main effects of these risk factors on brain development, we also looked at an interaction between family history and stress. So on the one hand, you could think individuals with a family history for disorder, when you add stress in, that's going to even further exacerbate the risk and lead to even greater changes in brain function. On the other hand, there's some studies that suggest that stress is actually a more critical component of the development of depression for individuals with no prior family history. And the thinking is that if you have no prior genetic risk for depression, those environmental inputs of stress are going to be particularly critical in terms of determining your risk. Whereas if you have this prior genetic loading, then maybe even mild levels of stressors could lead you to develop depression. So it might be less important if you have a family history. So we hypothesized that low-risk adolescents who had no family history for depression were experiencing relatively low levels of life stress would evidence decreases in amygdala reactivity during adolescence, consistent with that prior cross-sectional work. And then we hypothesized that high-risk adolescents, either due to a family history for depression or experiencing relatively high levels of life stress, would fail to show these typical declines in reactivity with age. And we also hypothesized, because our adolescents were on average younger than this peak age at onset at both of our waves, that we'd be able to see these changes prior to the onset of disorder. So because we had such a high-risk sample, we actually did have adolescents in our sample that had internalizing disorders. So about 30 in the family history positive group had an anxiety disorder at baseline. Nobody could have a depressive disorder at baseline. That was exclusion criteria. And then about 16 actually developed depression before the second wave of our study. So what we did was we first ran all analyses including all of these participants to get the full spectrum of behavior, and then we re-ran all analyses excluding participants with an internalizing disorder and controlling for depressive symptoms to see whether any changes in brain development that we saw still held for those individuals who had yet to go on to develop the disorder. Okay, so my postdoc advisor is Ahmad Hariri, so we use the Hariri hammer as our fMRI paradigm. And not surprisingly, so during this task, you see the trio of faces, and you have to select which of the two faces on the bottom row matches the face on the top row. And this has been commonly used as a good probe for amygdala reactivity. So in this version of the paradigm, we had blocks of either fearful face matching, which you see there, or angry face matching, and those were interleaved with our sensory motor control condition of shape matching. So adolescents were scanned twice. At the first wave, they were on average 13 years old, but they could range from 11 to 15. And we had 120 adolescents with a positive family history for depression and 112 with a negative family history. And then they were scanned again 2 years later. So at the second wave, they were on average 15 years old. And we had about 100 in each group for the second wave. So this is showing you the main effective task, face matching versus shape matching, at wave 1 and wave 2. So you can see that we got fairly consistent patterns of whole brain activation. And then in order to get an estimate of amygdala reactivity that I could enter into linear mixed models, I performed a conjunction analysis to identify all voxels in the amygdala that were significantly activated to the task at both waves of the study, and then extracted a mean beta parameter estimate of amygdala reactivity. So we got that for wave 1 and wave 2 for the left and right amygdala, and then for the contrast of angry faces versus shapes and fearful faces versus shapes. Okay, so what did we see? So for my first pass at this, I just looked at a main effect of family history for depression and looked at whether that affected the changes we saw in amygdala reactivity with age by evaluating a family history by age interaction. And I found significant effects specifically for left amygdala reactivity to fearful facial expressions. And this interaction was driven by the positive family history adolescents showing a significant increase in amygdala reactivity with age. Whereas the negative family history adolescents actually had a flat slope, so they showed no increases or decreases with age. I also, we reran these looking at change scores as our outcome instead of the linear mixed models to really drill down on within-person change across waves. And you get the same effect with change scores. So from wave 1 to wave 2, the positive family history adolescents are showing greater than expected increases in reactivity with time. Critically, when we remove those adolescents who had an internalizing disorder and control for depressive symptoms, these effects still hold and are still significant, suggesting that we see this even in adolescents who haven't yet developed disorder, and that this could represent a premorbid vulnerability for the subsequent development of depression. So next we looked at interactions with stress, and we tested effects of both childhood maltreatment as well as stressful life events in adolescence. And we only got significant effects for stressful life events in adolescence. So I will report those results here. And these are specifically stressful life events that are occurring in the year prior to the first scan. So for most participants, this is early to mid-adolescent stressful life events. And we got interactions between family history and age, stress and age, and family history and stress. So I'll walk you through what that meant in terms of differential patterns of development. So the red line are the adolescents who have a positive family history for depression who are experiencing relatively high life stress in adolescence. And they show the greatest increases in amygdala reactivity with age. The purple line are the family history